Okay, so protein. So whether we're eating a protein and it's like a complete protein or an incomplete protein, it doesn't matter. It's going to follow this same pathway. And the whole point is to take this protein, which is going to have a ton of amino acids bonded together, and break it down into its smallest form, which would be the singular amino acids. Okay? So starting with the mouth. So protein differs a bit from carbs and fats. So in the mouth, we don't have any salivary enzymes to start breaking down protein. So the only type of digestion that happens in our mouth for protein is mechanical, tearing it, breaking it into smaller pieces. So we're going to say here, no chemical digestion. But lots of mechanical digestion. Especially if we're eating animal fleshes. You know, we got to tear those a lot with our teeth before they're even swallowable. All right, so now once we hit the stomach, so this is where it really picks up its speed. So if you remember, what happens to carbohydrates, like the chemical digestion in carbohydrates when it hits the stomach? It stops, right? And so fats, there is an enzyme, a gastric lipase, that kind of starts to break the fats into little droplets, but it kind of separates from the rest of the kind because it's just not water soluble. But with protein, a ton happens here. So in your stomach, you've got your stomach lining, and it's got very, very specialized cells that are part of this lining. And so one of those cells are called parietal cells. And they release hydrochloric acid, or HCl. So this acid makes the pH of our stomach like near 1. It's crazy, crazy acidic. And so it's very separated from the rest of our bodies. If our stomachs were to rupture, that would be emergency room visit right away. So what this acid does to the protein is it denatures the protein. And so what this means, that's supposed to be in it. What this means is it starts that bond breaking. If you remember, proteins are, they have a primary structure, a secondary structure, a tertiary structure, and all of them, these structures are formed by random bonds that it has. So the primary structure is the chain amino acid bonded to amino acid. The secondary structure is the amino acid bonded to, or the hydrogen bonds that are happening. And the tertiary structure is the hydrogen and the sulfur bonds. So this acid is starting to denature all of those bonds and take that structure from being these more complicated tertiary and secondary structures and breaking them down as small as it can, okay? But it just kind of starts this process of denaturing. So additionally, you have some more cells that are in your stomach lining called chief cells. And so these cells will produce an enzyme called pepsinogen. Pepsinogen, however, alone is inactive, okay? And so what happens is it will come into contact with this hydrochloric acid and become active, and therefore become a different enzyme called pepsin. So pepsinogen in its inactive form comes in contact with this hydrochloric acid, giving us pepsin, which is active, okay? Same things going on here. So this pepsin is picking up where the hydrochloric acid is not, to keep breaking those structures down as small as it possibly can. Now, remember, digestion in the stomach lasts anywhere from like 4 to 6 hours. Whereas this is a real, slow process that's going on here. then over that time, the stomach will dose out bits of this chyme into the small intestines in amounts that are, actually gonna fit. Really, it just has to be small enough. So now we move to the small intestines after 4 to 6 hours. And again, pancreas, very, very important accessory organ in digestion. Pancreas pick up where the hydrochloric acid and pepsin have left off. And so it produces its own enzyme called protease. So here, pancreas release protease, okay? And so this protease, this enzyme, essentially breaks everything into almost its last form. So you end up with some singular amino acids here. You might end up with a dipeptide. Polypeptide is a multiple amino acids peptide bonded together. A dipeptide would be 2. And then you might end up with some tripeptides here. So this is what we've got at this point of the food entering the small intestines. You start getting these dipeptides, tripeptides, and single amino acids. Okay? So, let's move on to absorption. So now we've gotten all of this stuff broken down via these enzymes, starting with the hydrochloric acid and the pepsin in the stomach, proteases in the pancreas that get released into the small intestines. And so what we're left with in the lumen of the small intestines are these single amino acids, these dipeptides, or these tripeptides. And at this point, they can diffuse across the brush border of the enterocyte and start the absorption period. So let's say amino acid, di, and tris pass through brush border membrane into the enterocyte. And then one more enzyme comes into play here. So peptidase is now released inside of the enterocyte, or lives inside of the enterocyte, and it's gonna basically finish the job. So any di gets broken into its 2 single amino acids, and any tri gets broken into its 3 single amino acids. So after this point, peptidase, now we've got just the amino acids, okay? The potentially 20, right? Because we've got 20 that exist in our food. So now let's see, the enzyme peptidase finishes the job. All right. So these amino acids will pass through the membrane of the enterocyte and end up getting absorbed into that portal vein. You guys remember where the portal vein goes, right? It goes to where? The liver. You got it. Okay. So they enter the portal vein, they go through the portal circulatory system, and eventually into the liver. There is one situation that does not follow this pathway. Has anyone ever heard of branched chain amino acids? Anybody heard of the supplements of people taking BCAAs? Yeah. So branched chain amino acids are 3 essential amino acids that don't follow this pathway because of their structure. And so the branched chain means that it has, instead of having 1 carbon like that in the center, it's going to have 3 or more on its side chain. This is the branched chain part of a branched chain amino acid. The rest of them are just going to have that single carbon. And so which ones are these? Anybody know? Leucine is one. Yeah? No. You can supplement with that though. Anyone? So isoleucine and valine. So if you have your book, what page is it that has all the amino acids listed? Page 181. You'll see in the essential amino acid column, you'll see those 3, the leucine, isoleucine, and valine. And so these branched chain amino acids are found in food, and often people will supplement for them, or supplement them, or take them in a powder or something. So what happens here with BCAAs, instead of going into the portal vein, they enter our systemic circulatory system right away. They kind of bypass the liver, and they target muscle protein synthesis. They essentially go straight to the muscle. It's like almost they know their job. It's kind of funny. So if we have BCAAs, I'm just gonna say a B for B. It's gonna end up in systemic circulatory system. And they go straight to the muscles. So these 3 amino acids are basically the ones that are most stimulatory of muscle hypertrophy, and then also of protein synthesis of the muscle for recovery. So when would someone might want to take BCAAs as a supplement? Yeah, after working out. Yeah, someone's looking to get swole, looking for their beach bod, right? BCAAs might be might be your ticket there. So they bypass the portal system and liver. And they go straight to muscles. Okay. One in particular of these 3 is even more stimulatory, and that's leucine. So individuals that will take like whey powder as a supplement is heavy in leucine. All right. So let's, without this branched chain amino acid caveat here, let's go back to our more regular pathway. So the rest of them will travel to the liver, and they end up kind of in this, they call it the amino acid pool. I like to think of it as, I don't know, when you hear pool, I think of a swimming pool. But the pool is kind of like this area where they all just kind of hang out, ready for the liver to be like, I want you, you, and you, go over there, and I want you, and you, and you, and you, and you, and I want you to go over there. And so the liver's deciding where these all need to go, dependent on the need from the body. So all of these singular amino acids then get dumped into the liver amino acid pool and wait for direction. So, write that down. So amino acids enter the pool liver, like I said, is my favorite organ, and has a lot to do with everything else's function. This is just another one of its major jobs. So, for instance, say in this situation, someone is in a low energy availability. What might this liver prioritize these amino acids to do?
Maybe to
Yeah, how?
because they'll get broken down because they're still proteins.
What do they become? Glucose, good. So, in the situation of low energy availability, when I say energy, meaning the other macronutrients that generally produce ATP, amino acids can become glucose in the liver. Who remembers the name of this process? It's a G word. You got it, good. This is gluconeogenesis. Alright. So, this can happen daily to us if we go for a long period of time without eating. For instance, if we were to have dinner at 4:30, 5 o'clock, and nothing afterwards, go to bed and not wake up and eat breakfast till 8 a.m., that's a long period of time. We will deplete hepatic glycogen stores at that point. And so gluconeogenesis will start to happen until we eat. And so this isn't necessarily super detrimental in a short period of time or in these kind of quick scenarios. But in a chronic state of low energy availability, this is obviously going to start taking away from protein's major role, and that is for structure and protein synthesis, essentially. So say we are in a state of plenty of energy, we are going to, or the liver is going to decide that these amino acids are going to get sent to the cells that need them. You've got some red blood cells that are needing to be regenerated, they'll get some. If you've got some muscles that need to be built, they'll get some. If you just got the flu and you need some antibodies, that'll get some. And so this, the liver kind of distributes it dependent on the need. These needs could happen all at once, they could happen one at a time. It's just, we're always in need of some sort of structure or regeneration in our cells. And so this is just kind of always happening. So in a state of adequate energy, amino acids will enter whatever cell needs it and go through protein synthesis. Okay. And so our circulatory system is the highway that these amino acids travel from the liver to the cells.
So, normally when you go into where protein, like the cell needs protein, but if we don't have anything, then it has to do this first.
Yeah, because generating ATP, or essentially producing glucose for our central nervous system, is kind of a survival, or is a survival function. And so that's going to take precedent over anything else. Questions on this so far? You? Okay. So now let's talk about protein metabolism. All right, so these amino acids are now being distributed by the liver into the bloodstream and being sent to whatever cells it needs. And so now the cell is going to take these amino acids and then reassemble them into the specific protein that it needs. So if it's a muscle tissue that needs repair, this will be a certain message that the cell will get. if it's antibodies that need to be produced, those immune cells will get that message. And so depending on the message that's being sent and the amino acids that are being delivered, this is what's gonna dictate what type of protein structure or the sequence of those amino acids that cell will create. So imagine this, let's come up with a scenario. Let's say that this is a white blood cell, and we need to produce some antibodies to get rid of influenza A. It seems to be going around right now. So let's pretend that this cell is an immune cell, and we're trying to produce some structure, and that structure is an antibody, or the antibodies for it. So this should be a review if you had biology, or if you had A&P. So hopefully this won't be new information, but let's walk through it. So first of all, the cell is going to get a message that it needs to produce that structure, okay? Okay, and our example here is we need to produce antibodies. Okay. So this message will get sent to the nucleus of the cell, okay? What lives inside of the nucleus? The DNA, you got it. So all of our genes, all of our, basically the code for all of our structures and functions in our body. And so this message is going to pertain to a specific part of a specific strand of DNA, okay? And so, for instance, we're trying to create an antibody, we're not trying to create anything else, so that specific part of that DNA will then unwind from the double helix structure and expose that sequence of nucleotides. You guys remember what I'm talking about here? You've got the sugar, the deoxyribose, which is I would say if you're gonna look at it like a ladder, it's gonna be not the rungs, whatever you call this part, you know what I'm talking about. And then the rungs of the ladder are the nucleotide bases, okay? And the nucleic acids. So anyway, so here's my little RNA and DNA here. So the nucleus receives this message. And then the related gene sequence on the DNA unwinds and exposes itself. So also in our nucleus we have the RNA nucleic acids, and they will start to assemble a half strand, this ribose strand that matches the nucleotides that have been exposed from the DNA. So it's essentially creating a copy, right? A copy of this message. And so the reason this has to happen is because DNA cannot leave the nucleus, it's too large. And so the RNA just copies that one little sequence, that message, to take it out of the nucleus so that it can actually generate the structure. And so that is step 3. So then RNA assembles a copy of DNA sequence that's related to that message, and then the RNA strand will leave the nucleus. nucleus, you have these little pores all along the membrane. And that's how the RNA leaves the nucleus and enters the cytoplasm of the cell. So now this RNA strand that has this specific message, this specific code, is going to be outside of the nucleus and into the cytoplasm, and it's going to look for a ribosome. Who knows what structure the ribosomes are housed on inside? What organelle? Yes, good job. Did you have biology recently, or are you just really good at remembering that stuff? Okay. Cool. So then the RNA is going to find a ribosome that's sitting on the endoplasmic reticulum. And you guys know what a zip tie looks like? You put the end of a zip tie through the head of the zip tie, it kind of like zips through like that. That's what I picture when I picture an RNA going through a ribosome, okay? So this ribosome, here's an example of a ribosome, a nice drawing. This RNA is then going to go through the ribosome and kind of be read, imagining like the zip tie going through the head of the zip tie, just like that, okay? Also, floating around in cytoplasm are structures called tRNA, okay? So they look kind of like this. This is a tRNA, okay? So this RNA goes through the ribosome, and every 3 nucleotides that are on this RNA will have a complementary tRNA that will match up to it. These tRNA structures are also protein structures, which is another thing that your cells would need to produce from dietary protein or from amino acids. So tRNA floating around in the cytoplasm will find its complementary tides that will match up with this RNA. Also attached to the tRNA is an amino acid, okay? So you've got this tRNA, this amino acid. Imagine this is leucine, imagine this is valine, whatever the amino acid is. But this sequence of these nucleotides is going to dictate which amino acid goes next, and then which goes next, and which goes next. So as the RNA is going through the ribosome, the tRNA is matching up, and it will release this amino acid in the sequence along the nucleotides of the RNA. And those amino acids get bonded together with the peptide bond, and then you've got your first structure of a protein, the primary structure of that chain of amino acids. Does that make sense? Okay, I'll write it out here now. So RNA finds the ribosome on the on the ER reads the RNA, and then the tRNA finds its matching sequence. Releases its amino acid. And bonds it to the previous amino acids. So then you end up with this right here. Okay? Amino acid, peptide bond. Amino acid, peptide bond to the next amino acid. And so on. And you've got your primary structure. So if we were to look under, I don't know what it actually would look like, but an antibody looks kind of like that Y, and it has its specific little binding site to the specific antigen. But imagine that structure being this wound up tertiary structure of a protein along with all of the other parts and pieces that it needs. So that's kind of what we're creating right here. And there you go. So now, so let's talk about if we wouldn't be able to build the antibodies that we need to create the immunity to influenza A, right? Or we couldn't even create the tRNA that we needed to even read the RNA. Or maybe the ribosomes on the ER. And there's so many things that are essential to having this amino acid availability in all of them. So consuming too little protein or too little or not enough of the amino acids daily will end up in deficiencies. And this will end up in a loss of protein synthesis, and that applies to all of the functions the body has. And then there are some specific diseases. So your book, I want you to read this in your book. There's a couple of diseases, and this is these diseases are prevalent in impoverished countries, places that don't have food supply and are, usually have a lot of disease. And it's kind of like a starvation disease. And so these individuals, or these typically infants and children that will suffer from this, they will have improper brain development, obviously just malnutrition, heart tissue will begin to deteriorate, and usually they will die of some sort of infection because they don't have an immune system, or just organ failure because of the inability for those organs to develop. It's pretty serious, pretty sad, but it's kind of due to this protein, this macronutrient deficiency. So all those structures depend on it. So let's talk about consuming too much protein. Who here has heard that you should not eat too much protein? Why? What have you heard?
Hurting can mess up your kidneys.
Yeah. So here's why, okay? So research is kind of inconclusive, really, on whether this is actually detrimental or not. But here I'll explain why. So when your body processes or denatures these proteins, there's a byproduct of nitrogen. And so what happens to the nitrogen is it gets excreted out of your body through the filtration of your kidneys and out in your urine. And so these recommendations, the RDAs, are actually developed from studies where individuals' nitrogen balance was measured. So they would eat a given amount of protein, and then the individual's pee and stool would be measured for nitrogen. And if they were releasing more nitrogen than they were taking in, they were in a negative balance, and therefore they would need to take in more. And so these RDAs kind of come from that nitrogen balance research. However, this nitrogen balance research is got some, say, limitations, because you release nitrogen in sweat or any type of bodily fluid excretion, and so it's not an exact science. And so a lot of times people will negate the studies that have that nitrogen balance methodology because it's got some inadequacies. So this is why it's been said that consuming too much protein can damage your kidneys because of just the immense amount of nitrogen that you could be having to filter and then pee out. And they were saying that people that have renal disease, which is kidney disease, and individuals with other complications, diabetes being one of them that does affect their kidneys, high blood pressure, that they were to eat a low protein diet because of this reason. However, further research has shown that a low protein diet is not at all helpful, and all it does is create deficiencies. And so an individual should, in those cases with kidney problems, should eat the amount of protein that is recommended. However, what is too much? And I think that is the big question right now, because what the more current research is showing is that more is actually safe and beneficial, especially in situations for athletes and in situations for older adults and developing children. And so RDAs have been established for for those populations. And so I would assume, and this is just me assuming this, if you were to consume more than that RDA, then that could be seen as possibly a bad thing for your body. So what is too much? Well, it depends on who you are. Depends on your need, depends on your age, your physical activity, your health of your kidney. And so it's really, it's very individual. And so, yeah. The studies on what is too much is inconclusive, but they do say that individuals that do have kidney problems should not be on the upper end of that RDA. Does that make sense? It's like, what is too much? Well, it just depends. It's kind of hard to know. So, sticking with the RDAs and any research that comes from, say, ACSM or the ISSN for athletes is going to be a good range for you. And then you got to think about, obviously, I'll kind of just conclude this conversation, being mindful of your protein sources. So remember, until further research has really elucidated exactly the situation for saturated fats, the recommendations say to keep saturated fat less than 10% of your kcals. And what sources of food tend to have a lot of saturated fat or fatty animal products, right? And so just being mindful of how much you consume of those. And then obviously strive for all essential amino acids every day. If you're a vegan or vegetarian, then you would want to get those complementary proteins. Look for soy and quinoa. If you're vegetarian and do eat eggs and cheese and dairy products, then obviously you're going to get them from there. And if you do not, then you can get them from your animal products. And then in the situation of veganism and vegetarianism, there are a lot of vitamins that are only found in animal products or found in large amounts in animal products. Vitamin B12 is a biggie. It's only found in animal products. And so for individuals that are not eating animal products, vitamin B12 supplements are encouraged. And I think actually soy products do have vitamin B12. You would just want to make sure that you meet your RDA on that. And so it is a common vitamin deficiency. And B12 is pertinent to nervous system function. And so without it, people feel fatigued. They might get weird tingling in their hands and feet. They might have some cognitive and memory issues. So it's a very important vitamin. Vitamin C, vitamin D, or not vitamin C, vitamin D that's found in your dairy products. Calcium, iron that's found in meat. All of these are super important for athletes, especially that are, looking for protein synthesis just to maintain their fitness, and then individuals that are older adults that tend to lose bone mineral mass and bone mineral density and muscle mass, all that stuff. So vitamin D, iron, calcium, all of those vitamins that are found in large amounts in animal products, you want to just make sure that you're getting your source if you're not going to eat those animal products. And we will discuss these micronutrients in more detail after spring break. We're getting into the weeds on some vitamins and minerals at that point. So, but taking protein or taking animal products out of your diet, you just want to make sure that you are still getting these vitamins. That's primarily where they come. All right. Questions on protein?
Is this why they say, I guess I never made this connection in my head, but, severe alcoholics, or just alcoholics in general, they, bruise a lot more easily and don't heal as fast because it must be associated with this, right? Because they can't rebuild stuff physically.
Yeah, I think it might have to do with a behavioral thing mostly, but then I'm sure that there is some sort of dysfunction in the liver due to alcoholism. The ability to prioritize, and reorganize the amino acids to what you need. But I think what I read is that it's primarily a behavioral thing, that individuals that are alcoholic, tend to not even eat the nutrients that they need on a daily basis. Like it's a low energy availability, it's, usually vitamin and mineral deficiencies, just due to not eating enough.
So it's like stuff that happened maybe prior to it.
Yeah, so like having cirrhosis or other things, yeah. Anything else you guys have heard about protein and questions about? Alright. Cool. So, next topic, fluid and electrolytes. So start reading that chapter and we'll get started on that conversation on Thursday.